Project is about biosorption of heavy metal pollutants, lead and chromium using spirulina. Objective of the project is to study the absorption of industrial pollutants from aqueous solutions using spirulina platensis, to prepare the raw adsorbents for effective removal and characterization studies, to study the effect of various parameters such as initial concentration, temperature, contact time, and adsorbent loading on the removal in batch experimental studies, and to evaluate the results of the experimental studies done. The scope of the project aims at utilization of algae with high potential to adsorb heavy metals from wastewater, resulting in high quality reusable efficient water. Encouraging results were obtained from this project. Spirulina is a biomass blue-green microalgae that can be consumed by humans and other animals. The two species are Arthrospira platensis and Arthrospira maxima. In this project, Arthrospira platensis has been used. It is also used as a dietary supplement worldwide. Arthrospira platensis in powder form after harvesting. There are many methods for the removal of heavy metals. The biological approach is called as bioremediation. Bioremediation can be defined as any process that uses microorganisms, fungi, green plants or their enzymes to return the natural environment altered by the contaminants to its original condition. And the advantages of the bioremediation are low cost, high efficiency, minimization of chemical and biological sludge, regeneration of biosorbents and possibility of metal recovery. Biosorption and bioaccumulation comes under the category of bioremediation. The difference between the biosorption and bioaccumulation is bioaccumulation is defined as the phenomenon of living cells, whereas biosorption mechanisms are based on the use of dead biomass. The materials and methods used in this project. The metal salts used in this work are lead acetate and potassium dichromate as respective source of metal ions lead and chromium. Hydrochloric acid and sodium hydroxide were used for adjusting pH. The biosorbent material spirulina for the study were purchased from Offer, an NGO cultivating spirulina on commercial scale. The preparation of metal solutions. The heavy metal at various concentration is prepared synthetically. The lead solution and the chromium solution concentrations were 10 ppm, 20 ppm and 30 ppm. These are prepared and kept at respective conical flasks at room temperature. The batch biosorption experiment of metal were conducted with solutions prepared from metal salts. The working volume of metal solution is 100 ml taken in 250 ml conical flask. Biosorbent dosage is kept constant, which is 0.1 gram per 100 ml, and the biomass was added to the several flasks. The flasks were stirred at constant speed of 150 rpm at room temperature in a rotary shaker. Test samples were collected at regular interval of time and filtered using filter paper for the estimation of metal concentration. Experiments were carried out over a range of operating conditions. The biosorption capacity of spirulina affected by the range of operating condition is analyzed by UV visible spectrophotometer in this project. The results yielded by the lead removal. The results yielded by the chromium removal. Conclusion of the project. The bioabsorption capacity of Arthrospira platensis biomass was investigated for the removal of lead and chromium ions from aqueous solutions. The bioabsorption capacity of dry spirulina biomass was obviously affected by the operating conditions such as solution pH, contact time, biomass dosage and initial metal concentration. The lead removal percentage at the end of 120 minutes for the initial metal concentrations 10 ppm, 20 ppm and 30 ppm are 61.55%, 51.36% and 44.25% respectively. The chromium removal percentage at the end of 120 minutes for the initial metal concentrations 10 ppm, 20 ppm and 30 ppm are 83.8%, 73.75% and 59.57%. The optimum conditions for lead and chromium bioabsorption were pH 5 and contact time 120 minutes at room temperature. Furthermore, the bioabsorption capacity of spirulina is greater for chromium ions in comparison to lead ions. The overall result showed the dry spirulina biomass is an effective adsorbent that can be used for the removal of heavy metals, lead and chromium from wastewater.